We did a diagram like this the other day. We compared humans to ferns, or it was moss, I guess. So um, here we have the egg. You see how much bigger it is than the sperm. Um, but in order to get those, in order to go from regular two-end um, organisms like us to haploid end, you need a process called meiosis. And what that does, it produces these haploid sperm and egg, these gametes. And then when they come together, they can make the fertilized egg, a process known as fertilization. Oops. Corrected that on the fly real quick. And once you have fertilization, mitosis helps that embryo to grow very quickly into the adult form. And where does meiosis happen? It only happens in these limited areas in our body. In males, it's the testes, and females, it's the ovary. That's the only place where meiosis happens. Everywhere else, you have mitosis. And it makes it kind of confusing is that not only do you have meiosis occurring in the ovaries and testes, you also have mitosis happening in the earlier development of the sperm and the eggs. So you have both mitosis and meiosis in the testes and the ovaries. So we need to know the difference between mitosis and meiosis. And basically, you have the sister chromatids separating in mitosis. You're going from 2N to 2N, just making copies. So the sister chromatids are separated, and in meiosis, you have homologous chromosomes separated. Homologous chromosomes separated. And this is a pretty decent one, but I went ahead and drew an even better one right here. As you can see, mitosis, we got our chromosome here, it's been copied. This is the exact same as this, totally the same. And they get separated, these two new cells. And since these two chromatids are exactly alike, they're identical, these two new cells are identical. Pretty basic, although in reality we know it's a lot more complicated. But for our purposes here, we'll say that, or I will. Meiosis, more complicated. You have our homologous chromosomes lining up. Check this out. Homologous chromosomes lining up here. Here, like if this was mitosis, this chromosome would slide in right beneath it and it would get separated. But in meiosis, and remember, in the ovaries and the testes, it's different. Can you hear me a little better? That's me talking into the microphone. And when you have that separation of the homologous chromosomes, this is where you go from being 2N, now you're in, into just N, you're haploid. So this is, this is diploid here. This down here is haploid. You got just a single chromosome. That's kind of sloppy, I'm sorry. And then this process here looks just like mitosis essentially. This is meiosis 2. This is meiosis 1. So these two sister chromatids are separated. Down here, you notice you have 4 cells at the end. Here, you just have 2. So that's a nice difference you're going to want to note. And here we have, more developed diagram. One thing we want to definitely stress is this crossing over that happens in prophase 1. You have little bits of material from one chromosome popping off onto the other, and you can see that symbolized here by these different colors. So this is we talk about how we shuffle the deck in meiosis. Chromosome switch on. We saw that diagram comparing mice chromosomes to human chromosomes. Very similar from these pieces of chromosomes rubbing off, switching, through evolutionary time. You can get a lot of different combinations. Sometimes those combinations don't work. They're not going to be, passed on. That organism's going to die if it creates a change that creates, a challenging situation for that individual. But if it works, that organism will pass on those genes. Remember, you have, this is totally off topic, well, sort of, not really. We have over-reproduction. We do have over-reproduction of offspring for a lot of organisms. Way more will be produced than will be able to survive. Humans, we don't have that so much anymore. We see just 200 years ago, average female had 4, 5, 6, up to 10 offspring, and likelihood 1 or 2 would pass away, would die due to childhood diseases, things unforeseen, things that we, not unforeseen, things that we now fix very easily with vaccines and other treatments. So here we have, in this split, these homologous chromosomes. You're now going to haploid. This right here, up until that split, is diploid. Okay? In prophase, metaphase, anaphase, I assume that you know those terms, so make sure you review those if you don't know them. But pretty cool, you see these electron micrographic images of the condensing chromosomes, them lining up on the metaphase plate. Remember, they pull themselves along those spindle fibers to separate. And here we have meiosis 2, which basically is mitosis, like it says there. We have these identical chromosomes being pulled apart, or identical chromatids. And you count 1, 2, 3, 4. They're all N. Okay, they all have half the number of chromosomes. So these would eventually become sperm. And in the case of females in oogenesis, only one of them will turn into egg. The others are called polar bodies, and they'll just disappear. They're just there to keep track of those extra chromosomes, and one will conserve. We talked about non-disjunction. We see right here, this chromosome should be over here, but instead it sticks. And what that creates is an imbalance. We have these 2 eggs or sperm with an extra chromosome, and these ones don't have enough. Now, typically these not viable. Not missing one, that embryo won't develop. But in some cases, like in if you have where did my thing go? Okay. If you have, this happens chromosome 21 or 18, for some reason our body will accept an extra one of these chromosomes within reason. Remember, people with Down syndrome and Edwards syndrome have serious physiological problems that they need to contend with that you don't have to worry about typically if you have the normal number of chromosomes. So non-disjunction. This is something we did today actually in class. This is the 6th of March. It was a Wednesday, but you see as this mother age advanced, especially 40 seems to be a real impactful age where, I just messed this up, where you do have quite a big influx or increase in risk of Down syndrome. But remember, all sorts of, not just Down syndrome, but all sorts of complications occur at this later maternal age. But the relationship, the greater the age of the mother, the greater chance of trisomy 21. Okay, and we talked about the conditions, why so many women are compelled to have, children later in life. Social factors with work. They pay a huge price when they need to leave the job market or their career to have children that men don't have to face, but should face. Remember, mandatory paternity leave. That's, I think, will be the biggest thing that will impact the gender equality that we see in society right now. So a lot of times when you have that advanced maternal age, or if there's so the age of the mother, or if you have certain if the parents are suspected of having some genetic, disease like family history, like they have a lot of cystic fibrosis in their family, or a lot of Huntington's disease, or a lot of sickle cell, they might get genetic testing done just to see what the chances, or see if their infant will, or their child will have some of these diseases. So there's different ways you can go about sampling. Now they're really looking at some cells from the fetus do get into the mother's blood. They're thinking they can eventually test the mother's blood. Right now, from what I understand, it's not. They can't get enough cells to make a real accurate prediction. When you're trying to predict if your baby's going to have a disease, you want to be really sure. You don't want to have false positives or false negatives. So the best way to do that is actually go in and get a little sample of cells from inside the uterus. And there's one way you do chorionic villi sampling. There's these chorionic villi. Remember, villi are these little bumps that help to increase the surface area. So you have this efficient exchange in the placenta. Remember, we talked about placenta before. I'm going to change. So you're basically getting a sample of the placenta sucking out through this tube. And you can see this, is a risk. You're going into the uterus while the fetus is developing and sucking out a little bit of tissue. So that can be a problem. You also have amniocentesis, okay, where you actually put this needle through the abdominal wall into the fluid surrounding the fetus amnion, and therefore again getting a fetal cell sample. So there are complications. There can be infections, disruption to the, sorry how sloppy that is, to the placenta. Remember, anytime you're doing anything invasive or going into your tissues, you can damage your tissues. Oh, just for fun, if you look up here, you have a little cross section of the intestine, all these little bumps. Remember, those are villi, and they help to increase surface area. Okay, surface area is really important. Increase that surface area. Okay. And here you should do some drawings like I did, showing the first division and the second division. Here we have, and I use green for the mother, purple for the father, because I like Prince. Oh yeah. Purple Rain, that album changed my life when I was in, what was that, 6th, 7th grade. Wow, what an artist. Unfortunately, the current drug epidemic with opioids had a big effect on him. And he was prescribed painkillers because he had bad hits from all the performing he did on stage. So he probably got hooked from, being given those drugs by his doctor for all the operations he had to have. And those are very powerful, addictive drugs. So we have, so this were me. These would be the chromosome I got from my mother, and this would be my father's. And just it so happened that I had one of each separate. Now for humans, you'd have 23 pairs of these, obviously. I'm not going to draw that here. And then in these two, after meiosis 1, you see these two chromosomes are in this cell, these two are in this cell here. And at this point, we went from 2N to N. And then the second part, you see those chromosomes being separated, the sister chromosomes being separated. So eventually we have 4 cells, and they're all haploid.